All right, in this lesson, we're going to be doing the second section of notes for Chapter 7, the nervous system. We're going to be doing the anatomy of a neuron, neuron classification, impulse transmission, and I don't know if we'll get to reflexes or not. We'll see. So we've talked about the fact that the functional cell in the nervous system are neurons. They are the champions that do the thinking, that do the working, and they're specialized to be very highly reactive and respond very quickly and to be very specific. Neuron anatomy, they have two main parts. They have a cell body and they have processes. And we know that the cell body can also be called the soma. We probably have used that term before. And processes come in two forms. They can be either dendrites or axons. Dendrites send messages towards the cell body, and processes are going to, axons are going to send information away from the cell body. And a neuron can only have one axon. So the cell body is considered the metabolic center of the cell. It's where the nucleus is. It's where the majority of the cytoplasmic organelles are. We've talked about in class that neurons are amitotic, meaning that they do not go through the complete cell cycle. They do not complete mitosis. Something that also supports their amitotic nature is that they lack centrioles. And if you remember from Chapter 3, centrioles are the organelles that are going to create the mitotic spindle fibers that are going to pull chromosomes apart during anaphase, and so they don't have those, so they can't successfully go through mitosis. They have a lot of rough endoplasmic reticulum in them, and just like we learned back in Chapter 6 with the sarcoplasmic reticulum of the muscle cell, the rough endoplasmic reticulum, or the RUR of the neuron, is called the Nissl substance. Sometimes it's called the Nissl body. And the cytoskeleton is called neurofibrils. Okay, so again, these are like these specific sub-anatomical terms for the organelles of specific types of cells. I'm not sure if you can see my cursor or not, but I'm pointing on the purple thing. The large purple thing would be the cell body, and then you have the nucleus, the nucleolus. Is where ribosomes are going to be made. The Nissl substance is pointing to the rough endoplasmic reticulum, and a dendrite, so that would be bringing in information or picking up or sensing information, and information would be going towards the cell body. The axon is not shown in this picture. So here in this picture, I hope you can see my cursor. The yellow is showing the cell body or the soma. And then additionally, you have these dendrites that are also shown in yellow. There's one yellow axon leaving the cell body. This would be a multipolar neuron because it has more than two processes. This is also in the peripheral nervous system, and I know that because these little clear things are Schwann cells. That little black thing is the nucleus of the Schwann cell, and they're creating some myelin. And then in between the areas of myelination on the axon, you've got these little uncovered areas, and those are the nodes of Ranvier. If we go all the way down to the end, you have the axon terminal. So the axon is going to split into a bunch of axon terminals, which will then create a synapse or a meeting place with another neuron in this instance, and we know that lots of things happen at the synaptic cleft or the synapse between cells. So again, cell body, dendrites, one axon. These are Schwann cells that produce myelin, and then the unmyelinated regions along the axon are called the node of Ranvier. So we've talked about this already, but we have two main types of processes coming off of the cell body. Dendrites convey information towards the cell body. Axons convey information away from the cell body. A neuron can only have one axon. Myelin, so that's that like whitish fatty insulation that is created both in the central nervous system and the peripheral nervous system. And the central nervous system is created by the oligodendrocyte cells, which can create enough myelin to cover several neurons. And in the peripheral nervous system, Myelin is created by Schwann cells that can only cover a really small section of one axon, and so, or a piece of one axon, and so you're going to need multiple Schwann cells along the length of the axon to protect it. And what does myelin do? Well, it's there for protection, but it also increases the rate at which the action potential can spread down the length of the axon. So again, this to me looks like a very Dr. Seuss neuron. You have the lightish purple here would be the soma or the cell body. It's got several dendrites coming off of it. You've got the nucleus, and then the long process coming off of the cell body would be the axon. We see the Schwann cells that are making the myelin. The myelin in this picture is in red. You've got these black unmyelinated regions, which are the nodes of Ranvier. And at the end of the axon, it's going to spread into these little feet-like processes that we learned about in Chapter 6, which are the axon terminals. This is just some terminology, and we have discussed all of these terms in class, but it would be in your best interest to know them. Nuclei, and ganglia are the same things. They are cell bodies, but one is found in the central nervous system and the other one is found in the peripheral nervous system. Tracts and nerves are bundles of processes. A tract is found in the central nervous system. A nerve is found in the peripheral nervous system. White matter is myelinated regions in the CNS, and gray matter is unmyelinated regions in the CNS. And most of the time, your processes are going to be myelinated, and your nuclei are going to be unmyelinated. We can classify our neurons two different ways, either by their structures, so how many processes they have coming off of the cell body, or their functions, so what they're doing. And we know we have three overlapping functions. We've got sensory input, integration, and motor output. So our sensory neurons are responsible for picking up changes going on in and outside of the body and reporting that information to the central nervous system. The majority of our sensory neurons are going to be unipolar. Motor neurons, they are going to carry out whatever response has been decided by the central nervous system, and they are mostly going to be multipolar. And then interneurons, they're responsible for integration. They receive the sensory input, decipher it, and then make the decision about what to do. They are mostly multipolar as far as their structure goes, but there are a few bipolar interneurons, particularly in the eye. And again, this is just going through the multipolar, bipolar, and unipolar. That is referring to the number of processes coming off of the cell body or soma. So multipolar is, 3 or more, essentially. Bipolar is going to be 2, 1 dendrite, 1 axon, and then unipolar is just one process that is split. Part is going to be axon, part is going to be dendrite. So this picture right here is showing a bipolar neuron right here, and this is saying that this is an interneuron that will be doing integration. A unipolar neuron would be our sensory neuron. They're not really doing any thinking. They're just picking up information on the dendritic end and then sending it right along as an axon, okay? And then here we have a multipolar neuron. The majority of our neurons are multipolar, where they have multiple dendrites and then always one axon leaving the cell body. And this is just a stem cell. That's what the primordial cell means. So in terms of physiology, neurons have to be irritable, meaning that they don't have to be cranky, but they have to be willing to pick up some sort of change chemically, and they have to be able to conduct that change or that action potential onto other neurons or other cells, okay? I'm going to stop right here because the next thing is going to be, actually no, I'll keep going. Why not? I've got a few minutes left. We learned about this physiology in Chapter 6, and so I think it shouldn't be too hard right now. So at rest, a neuron is, the neural lemma, which is the plasma membrane of a nerve cell, is going to be polarized, meaning that it's going to be slightly negative on the inside, slightly positive on the outside. There's an abundance of sodium on the outside just waiting to get in, and an abundance of potassium on the inside of the cell just waiting to get out, okay? When the dendrites sense some sort of change, it's either, sent to them because of a neurotransmitter or a receptor is picking up something. What happens is that this causes, in just a very localized area, sodium to rush in the cell. Just like you learned about at the neuromuscular junction, just at one specific area, sodium starts to rush into the cell until we reach that threshold potential. Once we reach that threshold potential, okay, then voltage-gated sodium channels are going to open along the entire length of the neuron. So it's going to go through the cell body, all the way down the axon to the axon terminal. Once this electrical current reaches the axon terminal, then those calcium channels are going to be opened, neurotransmitters are going to be released, and then the cycle happens all over again, okay? But in order for that neuron to send the action potential again, okay, or be able to pick up another stimulus, it has to repolarize, okay? So we have to go through that polarized depolarization till we reach the threshold potential, then an action potential fires, which is an all or none response that travels the entire length of the neural lemma. Once it reaches the, axon terminal, then it's going to stimulate neurotransmitters to be released. But before that neuron can send another signal, it's got to repolarize. So it depolarizes by sodium rushing into the cell, coming to a positive level, and then it's going to repolarize because potassium is leaving the cell, causing it to become negative again. Okay? At the synaptic cleft, or when you get down to the axon terminal, different neurotransmitters are going to be released. So calcium comes in. If you guys remember from chapter 6, the action potential causes voltage-gated calcium channels to open. Calcium causes the exocytosis of different vesicles that contain various neurotransmitters. We learned about acetylcholine in the muscular system, but you probably have heard of serotonin, dopamine. Okay, those are all neurotransmitters that are going to cause different things to happen. So this is showing multipolar neuron to multipolar neuron, and it's zeroing in. You see the little red square here. It's looking at this axon terminal dendrite meeting, okay? So the nerve impulse is coming down, the neurotransmitters are being released. It says NT released. The neurotransmitter moves down and binds onto receptors onto the dendrite of another cell. Those are ligand-gated channels. They're going to open up. They're going to cause sodium to rush in at that place, and the whole thing's going to happen again, where this polarized cell is going to become locally depolarized and then, reaches action potential and then move along its way again. And again, this is showing a synapse, so axon terminal, and then this is the postsynaptic cell, which is saying it's the dendrite of a second neuron. So this would be a neuron to a neuron. Last chapter, our postsynaptic cell was a muscle cell. This chapter, it's showing neuron to neuron. And I am going to stop here and not do reflexes. All right, so like I said, I do think that Chapter 6 set us up really well to understand the physiology of Chapter 7. We're still dealing with that polarize, depolarize, repolarize, sodium coming in, potassium going out, axon terminal, that space between our neuron to neuron, which is the synaptic cleft. Neurotransmitters are going to have to get secreted into the synaptic cleft to end up propagating or sending on the action potential to further neurons, and then, whatever the eventual cell is going to be, a muscle cell, a stomach cell, an eye cell, what have you. Alrighty, that is it for this section.